friends, welcome back in another video tutorial from microbiology with Chagdik. Welcome back in another revision class of methods in molecular cloning. So we are just recapitulating our concepts regarding gene transfer. So in our previous video we have discussed about some gene transfer method, and in that video we are also going to start to discussing our another gene transfer methods. So our first installment is Lipoplex mediated transfection and endocytosis. So it is another kind of gene transfer method that is commonly used. So here our first goal will be the Lipoplex formation that can be easily done with the help of liposome, which is positively charged, and with the help of DNA. Basically in the Lipoplex the DNA is enveloped by the liposome, as you can see, and as lipoplex is positively charged, it can be easily received by the host cell via the process called endocytosis. Okay, and as it enters inside the host cell via endocytosis, there will be a formation of endosome. Right now the next step is very important. There can be two possible options. Okay, if the lipoplex inside the endosome along with the DNA molecule, mainly the DNA molecule, can manage to escape from the endosome before the lysosomal pathway triggers, only then the DNA can survive inside the host cell and cloned or expressed. Otherwise, if lysosomal pathway triggers before the escape of DNA from endosome, then DNA will be simply degraded with the help of lysosomal pathway. So this is the step that mainly determines the efficiency of transformation process. So that is all about lipoplex mediated transfection and endocytosis. Now there are another problem that we are going to face, that is target specific transfer of gene cannot be performed too specifically with the help of this lipoplex mediated transformation. Why? Because most of the cell membrane, we all know, are made up of same components, right? And lipoplex is also itself made up of bilayer. So there is no specificity that can be used to insert our gene of interest in a target cell. So that is important and most biggest disadvantage of lipoplex mediated transfection and endocytosis. Although nowadays some ligand linked lipoplex are used to mediate the transfer target cell specific. Okay, so in terms of specificity there is some problem in case of lipoplex mediated transfection and endocytosis. Now let's discuss about the advantage. The first advantage is it is economical. Okay, the second advantage is efficient transfer of DNA can be done inside the cell in a culture dish with the help of liposomal mediated packaging and endocytosis. And here the delivery of DNA is with minimal toxicity. Okay, because as you can see there is no formation of pore. So except DNA molecule with the lipoplex, there is no too much chance to enter any other undesirable molecules, right, that may present in the cell culture. So that is another advantage that important, and it is easy to perform and required only some minimal steps, and it can be easily adapted for high throughput system. So it can be used for modern era gene transfer method. So that is the advantages, and there are some disadvantages too along with the method going on in case of specificity. So another disadvantage is it is not applicable to all kind of cell types. Okay, so this lipoplex mediated gene transformation and endocytosis fails to transfer gene in case of those cell lines that contains lipid. So that are the disadvantages that you need to know. So what is the overall summary? Summary is lipoplex mediated gene transformation is a type of gene transformation which involves the formation of lipoplex, and it contains two components. One is positively charged liposome, another one is negatively charged DNA. This lipoplex can be easily received by the host with the help of a pathway called endocytosis, and inside the cell they become enveloped with the endosome. And now if the DNA can escape from the endosome before the lysosomal pathway triggers, then only the DNA can survive, otherwise not. And then the rest of the process is same. Disadvantages are lipoplex mediated gene transformation cannot deliver our gene of interest specifically to the target cell. Why? Because most of the cell membrane is composed of same components, and lipoplex is also itself a bilayer, so there is no thing present to determine the specificity of the transfer. Okay, so for solution of that problem, nowadays ligand linked lipoplex are used to mediate the specific gene target inside by using lipoplex mediated transformation and endocytosis. And transfixion of gene with the help of lipoplex mediated transformation and endocytosis is failed in case of those cell lines that have lipid. Okay, so these are another kind of disadvantages. And most important and biggest advantage is here the delivery of DNA is with less toxicity. Okay, as there is no kind of formation. Now let's move to the Gengar method. That is our last gene transformation procedure for now. So Gengar method is another kind of gene transformation method that is commonly used for transferring our gene of interest in case of mainly surface cell or those cells which are exposed. Okay, so for performing gene gun we require such that apparatus. Okay, and this apparatus contains the same principle as a normal gun. Here the pulling force is a pulse of helium, and here some micro projectiles are used, which is nothing but gold and tungsten coated recombinant DNA molecules, sorry for that. And with the help of full of force, these micro projectiles, that means gold and tungsten coated recombinant DNA molecules, will be transferred to the host cell. Okay, and once they penetrate inside the cell, okay, once they penetrated inside the cell, due to the reducing environment inside the cell, the DNA is separated from the gold and tungsten, and as gold and tungsten is not too much harmful for the cell, so we can easily use this Gengar method for our gene transformation for our host inside our host cell. Okay, so that is the Gengar method. So what are the advantages of Gengar method? As it using a too much amount of force, there is no matter that whether what kind of architecture the cell possesses, either it contains cell wall or not, we don't need to bother about that when we are using Gengar. Why? Because here we are using too much force for transferring our micro projectiles, that is gold or tungsten embedded recombinant DNA, so they will simply penetrate the cell at any cost. Okay, and as a result we don't need to bother about the penetration. And what are the disadvantages? disadvantage is during the internal organ drug delivery, we cannot use that Gengar method. We can only use the Gengar method for those cells which are present in the surface or which are exposed. That are the disadvantages. So we have discussed about various kind of gene transformation procedure. We have started with the CaCl2 heat shock method, then we have discussed electroporation method, then we have discussed the micro injection method, then we have discussed today the lipoplex mediated packaging and endocytosis or fusion, and in the last one we have discussed about the Gengar. In the next video, we are going to discuss about the Agarobacterium tumefaciens mediated gene transformation. Although that video, I am not going to complete Agarobacterium tumefaciens mediated gene transfer in one video. I will separate it into 5 or 6 video, because that is the important part in that gene transformation chapter. Okay, so till then, bye bye.